Okay, hi, I'm Krista. I'm an editor at Molecular Cell, and I'm here with Houra Merrikh, who is a professor in the Department of Biochemistry at Vanderbilt, and her lab works on DNA replication and transcription replication conflicts, and also the evolution of bacterial resistance. So thanks for joining me today. I guess I'll just start by asking you to tell us a little bit about the two lines of research that you have in your lab and maybe the connections between them.
Yeah. So it's been a road that's, no pun intended, actually, an evolution of our research. So we were mostly interested in DNA replication and how transcription interrupts DNA replication. And we chose to study this in bacteria because the orientation of genes actually matter. So in bacteria, you have a single origin. So it was really easy for us to start looking at that. And we happened to, really early on, actually, in my lab, determine that depending on the orientation of the gene, transcription becomes more or less mutagenic. So then, we became curious, like, how does this work? And so in the, follow-up publications, we identified some factors that seem to be responsible for the accelerated evolution of genes that actually are transcribed head-on to replication. So it's like a car crash, you have these head-on collisions and there's an increased amount of mutations, and that depends, of course, on the level of transcription. I mean, transcription has always been known to be mutagenic too, but why is still, but I think we're getting somewhere with that. So that was sort of the beginning, and then, I wanted to study more, how do these factors impact evolution, and, how do they work? And so we wanted to simply just study that process. And when we thought about, what's the best way to study evolution, we thought, okay, antibiotics, great. You can kill cells, challenge them, and see how they evolve and adapt. So that's how we got into antibiotics. And we started studying these factors. And then some interesting events happened. So I was never really in the field to study clinical kind of aspects of things. But I happened to end up with a connection with the Gates Foundation, who is, are very interested in clinical problems. And there I learned a lot more about what a challenge antibiotic resistance is in the world. And I started thinking about that. We had these tools, and I realized that all we're doing to deal with this problem is making new antibiotics. And so this is the second part of the lab. And so that's the connection, and I'll tell you more about the second part, but the connection was studying the very basic biology of how cells replicate DNA and mutations arise. And that really led us to these sort of critical things that then led to the evolution of the lab to study antibiotic resistance development. So I realized that, there was this moment when I was hearing about data on how, all these children are dying, and, in different parts of the world that we don't really pay attention to enough. And it really made me feel almost responsible to go after the problem. It became kind of like an emotional thing, like I got to do something. So I started thinking about that and realized, we're not doing anything new. And so if you look at the timeline of, resistance development, you see that new antibiotics are being made all the time. And every single time, resistance arises. So I was like, we need something totally different. And at that time, I wasn't connecting the evolution part. I was just thinking, we need a new solution to this problem. And so somehow, it just one day occurred to me that, you know what? The only way to deal with the problem is to stop evolution and to reduce mutation rates. And at first, when I talked about this, people thought it was a little bit out there. But I knew we had these factors that accelerate evolution. So I just thought to myself, well, what if we disrupted these factors and created, some tool that we could use during treatment of infections that could reduce mutation rates and block mutagenesis mechanisms, and through that actually prevent drug resistance development. So that's really how it all started. And interestingly enough, we found that what used to be thought as a repair factor, this protein called MFD, is actually accelerating evolution, not only just in terms of like gene orientation and collisions, but just generally across the genome. So, feel free to interrupt me because I could talk about this all day. This is what I think about. So suddenly this repair factor, DNA repair factor, that has been studied for years biochemically, we started looking at it in cells without external DNA damage, which was the main thing that's actually different between what we've done and what other people have done. And when we looked at it, we realized it's actually a mutagenic factor. So from there, we learned some things. When we genetically manipulated and got rid of the gene that produces this protein, the cells couldn't evolve very well anymore. I mean, we would do these evolution assays, and we had to stop the experiment because the cells would become so resistant that now all we were doing was adding reagent. We couldn't add any more antibiotics. But when we didn't have this protein, they didn't evolve. So it was just a huge difference. So that's where it started, but then I realized, okay, like this is potentially doable, so now we need an anti-evolution drug. I got a little too ambitious maybe. But anyway, so we did, some in vivo screens in bacteria, and we looked for novel compounds that can get into bacterial cells and inhibit this particular protein. So we set up a system that was specifically isolating novel compounds that could inhibit this protein, with the idea that if we found something, we could then later, through medicinal chemistry, develop it for use in the clinic and potentially stop resistance development by inhibiting evolution.
Oh, wow. So you would use that in combination with antibiotics to keep them working longer.
Yeah.
That's interesting.
So, yeah, so we did that, and it's been a 7-year-long process, but I'm happy to say that we have found some novel compounds that actually inhibit, this particular factor, MFD, and actually inhibit evolution across totally like highly divergent bacteria. I mean, I'm talking about like Staph aureus, Pseudomonas, you name it. Everything we've looked at, it works, and the protein is highly conserved, so it's actually not that surprising that one drug would work in all these different bacteria, and that's really important in my opinion because what you want is a single drug.
Yeah.
We don't want to make 50 drugs, right? So anyway, so right now we have the story put together that hopefully we're going to be submitting for publication next week or two. But then we started asking some more questions. So the part of the lab that's working on replication transcription conflicts, that's still going on. And we've been very interested in R loops lately. They're very popular, but this RNA, DNA hybrids, and the single-stranded DNA. And we've come up with some new methodology to look at, how cells deal with this particular thing, because we found that, well, it wasn't just us, also another group, that these structures are a big problem during conflicts. So we're studying that right now. We have a bunch of new proteins that seem to be involved in these R loops and their formation, and there's a lot of interesting stuff. I mean, some of them seem to be toxic. That's very strange. So we're looking at all that stuff. It's really cool. But another thing that we've been working on that was really a cross between, it's sort of a bridge between the two sides, is actually what I'm going to talk about during this conference. And that was sort of, looking at nucleotide excision repair. So MFD is a protein that has been thought to be involved. In fact, but now there's some new papers saying that it may be doing something slightly different in nucleotide excision repair. So when you have a bulky lesion on DNA, this protein will recognize stalled RNA polymerases during transcription. And when it stalls, when the RNA polymerase stalls, this protein recognizes it and it removes it. And this is somehow involved with nucleotide excision repair. Now, that's been studied forever, decades and decades, right? But always under conditions, I shouldn't say always, mostly under conditions of exogenous damage, which is usually UV damage. Now, you have to realize that in bacteria, at least, UV damage is not something that the bugs see too often, so it's a bit, it's not exactly necessarily the right thing to look at. But in any case, we study everything without exogenous damage. So we started looking at nucleotide excision repair and MFD and their, relationship with each other. And honestly, kind of shockingly, we found that nucleotide excision repair is mutagenic. So it's not just MFD, which recognizes, transcription that's stalled. It's also just the entire process of nucleotide excision repair. And when we've done, we did a lot of bacterial genetics. That's my talk's going to be all bacterial genetics. All of these things are in the same pathway. So what this is telling us, and this is sort of another story that we're sort of wrapping up, is that nucleotide excision repair is pretty much almost always happening through transcription. And it's always mutagenic. So it's not just MFD. It's everything downstream too. So this idea that there's nucleotide excision repair that happens outside of transcription, I'm not sure that's true. Maybe sometimes, but what we see is this. So that's kind of the gist of all of it. I think I've sort of covered most of it. The only other sort of new angle that we're studying in our lab is now doing the same thing, basically, during cancer treatment. So there are a lot of new chemotherapy agents or different therapies generally coming out, but there are no cancer cells that are immune to mutagenesis and evolution. And so we have the same problem there. You always have this drug resistance problem, right? So the lab, that actually branch of the lab has slowly become a drug resistance branch. So in reality, we are studying drug resistance and how to stop that, but with the angle that really hasn't been explored, and that's reducing mutagenesis and stopping evolution. So that's kind of the big picture of everything.
Yeah, that's fascinating. I guess on the cancer side, that seems a little different than antibiotics. How would you use those therapeutics to specifically target cancer cells over normal cells?
Well, they don't exist yet. So that's what
works.
You have therapies, treat cancer cells. What we want to do is we have a pretty specific idea of where to look, but find the therapeutic that also slows evolution enough that, cancer therapies are more effective. And that would require an anti-evolution drug as well. So that's, we're directly looking for that right now.
Yeah, that's an interesting approach.
It's a very new project because I've always studied bacteria, so for us it's a bit new, but our department, so Vanderbilt Biochemistry, we have, I don't know if you've looked at the program, but we have multiple talks from just our department. So we have a great group of people, and we have a huge cancer center, so there are so many different people there that we were able to collaborate with that are helping us, you know, with this project. And, you know, I'm excited to see where it goes.
Yeah, that's really exciting.
I don't know if it's going to work, but it worked in bacteria. I mean, we're looking at such fundamental processes that, like, to me, you know, should be conserved from bacteria. I mean, and that's the thing, is if you look at the original papers, transcription has always been mutagenic, and it's been shown in bacteria, in yeast, in mammalian cells. Why? Right? That's the question. So I think that what we've been studying with this MFD protein and nucleotide excision repair, all of that, I think under normal conditions, when you don't mess around with the cells, they might actually be accelerating evolution, and that's the point. So if that's true, this is conserved, and we should be able to target it. So
yeah, that's a really interesting point. I guess we'll hear more about the nucleotide excision repair in your talk.
Yes.
So you think that's the mechanism by which transcription is promoting mutagenesis, or multiple mechanisms?
Honestly, yeah. And I may or may not have time to talk about it, but I think it all goes back to oxidative damage. So we've been able to, we've seen now in the lab, depending on, I have to time my talks. I don't know. But what we can see is evolution just stops in its tracks, you know, when we get rid of oxidative damage. And interestingly enough, so far, when we look at cells that don't have MFD or some of these nucleotide excision repair proteins, getting rid of oxidative damage doesn't change anything. I mean, these cells don't really evolve, but if you let it go forever, eventually they start getting mutations randomly, right? The replication fork will make mistakes.
Yeah.
All of what we're studying is separate from replication. This is all transcription. Doesn't matter if the cells are growing or not. So eventually, if you wait long enough and you start seeing some evolution, you can still get rid of oxidative stress. And there, we don't see a decrease. But in wild-type cells, we get rid of oxidative damage. I mean, they can't evolve. Now, the problem is, in human cells and in cancer, you don't want to get rid of oxidative stress, because that's how cells fight, like, infections. Let's talk about infections. During infections, you need macrophages to attack the bacteria with oxidative damage. So the answer is not, let's, you know, add a whole bunch of antioxidants and reduce the possibility of these bacteria becoming resistant to the antibiotic. That is not good. We don't want to do that.
Yeah.
We need something very specific in the bacteria that allows the host cells, like macrophages, to still kill the bacteria, but not allow the bacteria to actually develop resistance. So that's the goal.
Yeah, that's really exciting. A different approach than just continuing to develop new antibiotics, like you said.
I mean, we can keep doing that, and we should.
Both.
It's good, but we need something. I mean, at this point, we know too much. We should be coming up with some, you know, new ways of dealing with these problems.
Yeah.
So yeah, I mean, so I've been focused on bacteria, but the fundamentals are, you know, it's no different for me. Cancer is a great. But the bacteria has been a great model. Like, we learned all this stuff, right? So hopefully we can expand it beyond that, and hopefully, you know, this first lead compound, which needs to be developed, you know, you never find your clinically useful compound when you do your initial study. We have the lead compound that's very effective. So this is the first step, and we have a few more that we're interested in studying, and we'll continue to do that in bacteria, but we like to also expand the concept.
Yeah, that's interesting. It's kind of neat. I think your basic science background and, like, expertise in replication probably helped you to bring sort of a different perspective to studying antibiotics. Do you feel like that's true for you and your trainees, that like basic science brings a different perspective to the clinical work? Yeah,
I mean, I think there has been a disconnect almost, you know. And we study, like, and I did my PhD with Susan Lovett, And there we studied DNA repair. She was studying mutagenesis, and she laughs at me now. She's like, "You were in my lab, you never studied mutagenesis."
Yeah.
No, but there's been a disconnect where we're learning how mutations come up and how cells get rid of mutations and all of that. But then in the clinic, all people do is just say, "Oh, here's a mutation that causes resistance." Well, we know how to like play with this, so let's connect them. So it's kind of a big undertaking, but go big or go home.
Yeah, that's fascinating. No, that has the potential to be really impactful. So that sounds, a good question to ask.
So anyways, yeah, so I mean, that's basically what we're doing, trying to do.
Wonderful.
Everything is difficult, of course, for everybody.
Yeah.
But yeah.
Well, awesome. Thanks for chatting with me today. That was great.
Yeah. Sorry, I feel like I talked a lot, but
no.
If you ask us scientists about what they're doing, we're usually excited. So we have a lot to say.
Yeah.
But anyways, it was good talking to you as well.
Nice talking to you too. Thanks.